same tumor at the same time to be confirmation right?
Same
possible?
parameters from the original tumor,
Yeah.
for the primary tumor,
Yeah.
and after treatment,
Yeah.
it's the same pattern.
Oh.
Therefore, very, very expensive.
Oh, okay.
And that's the reason.
Yeah.
It's very, very expensive.
The problem with the tumor is that we say this enzyme or this marker is used for a prognostic for tumors, but then we suddenly discover that this is also correlated to infection or other disease. So every time we found that what we think it is specific for this tumor is not specific, because tumor changes its nature, and also we need more specific tumor, more specific markers for each tumor.
Exact. I told you yesterday, daily new parameters are found. And so it's not possible to make analysis from 100 or even more parameters per each tumor. It's not possible. Nobody can pay it. And therefore we focused on P53, BCL2, the cascade 3, BAC, and cytokeratin. So 10 parameters which are involved mostly in this tumor progression. Otherwise, nobody can do it. So mostly I'm alone in my lab, and I have to switch to what's useful. What is useful?
Besides, sometimes you have to depend on the, like, if it is supported by the government for insurance or not, sometimes the patient he can't pay, right? I don't know, how about Germany? Does the government support these?
Who covers the expenses of
Who covers
this?
it? It's really expensive.
yes.
In the report.
Is
Because you know, most of centers here, they are even the...
no, it's not research
Yes,
at this moment.
yes, it's clinical
Yes,
service.
It's clinical service,
Yes,
and the insurance,
covers it.
so most of them. But not each parameter.
So in other words, at least DNA cell cycle ploidy analysis, flow cytometric analysis is now considered to be as mainstream diagnostic
Exact.
test.
Compared to histology.
So it is required,
Yeah.
therefore the insurance will
Yeah.
pay for it.
It's a basic for insurance. And if
Yeah, remember here we tried to talk to the oncologists and try to lecture them, but no, there was no real interest from them to go this route.
for example, P53, it's also paid by the insurance. BCL2, about 5 or 6 parameters which will be paid, but not more.
We have
Dr. Abdul Halim is, in more molecular diagnostics and molecular analysis. Question is, in tumors, is there a move towards, trying to do, let's say, DNA or RNA analysis to try and distinguish different entities and
Now tumors, they are not classified as by their origin, or this is
Yes.
a lung tumor or
Yeah.
breast tumor.
Yeah.
Now we're talking about molecular molecular classification
Yeah.
for
Yeah.
tumor. This is,
Yeah.
and they could go for analysis of the genes. Actually,
Yeah.
they go for certain mutation like BRCA1, BRCA2, and
Yeah.
breast cancer and so on. And this is, I think this is a
For
future.
some, they're known, but I mean, generally,
Yeah, not for all.
is there any work towards trying to, like this work with flow cytometry, trying to find parameters that would further subdivide, you know, tumors into subclasses that would be consistent with better or worse outcome? Is there any kind of molecular
Yeah.
attempts to do
Yeah,
that
This
kind of
this is
diagnosis?
molecular.
Yeah.
No, yeah, okay, but I mean,
It's
Gene,
not about
you
the,
know,
around
I'm
the DNA
talking-
level.
I'm talking at the DNA or RNA level, you know,
Yes, there are an effort in this direction.
yeah,
There's still a long way to go.
yeah.
This is a part of
Yeah.
molecular
Yes, yes.
biology. Also, this is parameter.
Yeah.
The nuclear parameters as RB, for example, or
Yeah.
P53. It's localized in nuclei. BCL2, it's a bug system. Cascade, it's localized in cytoplasm,
Yeah,
especially in mitochondria. And this is molecular biology.
yeah, but all of these are proteins, and you're right?
It's
And you're detecting them with the,
most are proteins.
yeah.
But
I'm thinking of either DNA or RNA expression, you know.
yeah, and MYC-N, it's an oncogene,
Yes.
and you can remove it, and you can make from the removed MYC-N and can it used for testing in cytometry. Not as proteins, but it's an oncogene. And this gene you can cut from the chromosome,
And how, the question, how do you label it when you look at it? Do you label it with a complementary DNA, or do you label it with an antibody, or how?
This proteins with an antibody.
With antibody, yes.
The antibody
Yes.
and the color.
Yes.
The color fits or PE or
Yes.
whatever.
Yes.
Short break, then we have to proceed with the other lecture because
شو other lecture عندكم lecture
إحنا
ثانية؟
عندنا lecture.
وين؟
هنا
ما عندي خبر، أنا بعرف إنه في واحد.
I speak